In our last session, dedicated to mutations and DNA repair, we will learn about the mechanism of DNA repair in human cells, and we will also have a closer look at certain genetic disorders that result from mutations at gene level. DNA repair is a mechanism which is quite vital to the survival of the species. DNA has the intrinsic capability to repair itself, unlike RNA, because there is always an extra copy present. Again, remember that our DNA is double-stranded and genes are actually present on both strands. Therefore, if mutation occurs only on one DNA strand, the intact copy can be used by the cell as a template. Many errors in DNA can be actually recognized by the cell, and they can be corrected by two distinct mechanisms referred to as homology-directed repair, shortly HDR, and non-homologous end joining, short NHEJ. Homology-directed repair is extremely important for suppressing the formation of cancer cells because mutations occur on a daily basis in many of our cells. However, most of them are recognized, and I hope that you remember that DNA is actually controlled and checked during certain checkpoints in the cell cycle. The cell cycle does not progress if DNA is found to be faulty. This is actually also the place during the cell cycle where DNA repair takes place. HDR is the most important mechanism of DNA repair, and it maintains genomic stability by repairing broken DNA strands. It is also assumed to be error-free because it uses the undamaged intact template which is present on the second DNA strand. In contrast, non-homologous end joining is an imperfect repair system. It inserts or deletes base pairs and creates a frame shift. It is also prone to exon skipping and disrupts gene translation. So, those are the two types of DNA repair mechanisms you need to keep in mind because they are going to be fairly important for our lecture dedicated to gene therapy in the springtime. So, after we have learned the basics of DNA repair, let's have a look what happens when gene mutations cannot be repaired. Results of mutations can actually lead to many different disorders, and a very good and classic example of a genetic disorder resulting from single gene mutation is sickle cell anemia. Sickle cell anemia is a serious inherited blood disorder, and in England about 250,000 people are affected by the disorder. The genetic cause is a point mutation in one particular codon in the hemoglobin gene. In the wild-type hemoglobin DNA, one codon is CTT. This results in a production of an mRNA with the following sequence, GAA. This then produces glutamine in the normal hemoglobin protein. And this is extremely important protein in oxygen transport in the blood. In carriers of the mutation, in patients with sickle cell anemia, this triplet, however, is mutated. Instead of T in a healthy wild-type hemoglobin DNA, there is an A. This results to a different in a different mRNA, and finally, valine is inserted into the protein in sickle cell hemoglobin. At cellular level, this actually leads to quite few changes in the blood cells. In normal cells, healthy hemoglobin is present, and we have this flexible and disc-like shape. However, in sickle cell anemia, presence of mutated hemoglobin changes the appearance of the cell. They look like sickles and become rigid, therefore blocking several blood vessels. Those abnormal cells are unable to move as easily as normal blood cells and result in tissue and organ damage, and also in episodes of severe pain. Another example of a genetic disorder which is a result of point mutation is cystic fibrosis. Patients with cystic fibrosis have several symptoms affecting the lungs and digestive system because those organs are become clogged with thick and very sticky mucus. The frequency of cystic fibrosis is actually fairly high because it occurs in 1 in every 2,500 individuals. It is caused by a mutation in the so-called CFTR gene. Healthy CFTR gene normally creates a protein that regulates level of sodium and chloride in the cells. If this gene is mutated, it results in build-up of a thick and sticky mucus. In the healthy CFTR sequence, we have the following triplet present at this very position in the DNA. We have a CTT. However, this whole triplet is usually deleted in patients with cystic fibrosis. So we have a deletion of the CTT gene. Another example of genetic disorder, which is a result of gene mutation, is a disorder called neurofibromatosis. It affects 1 in 3,000 births and produces tumors that grow along nerves in the periphery of the body. And at genetic level, this disorder is a result of a mutation or a deletion of the so-called neurofibromin gene. The neurofibromin gene is a negative regulator of the RAS protein. I hope that some of you remember that RAS is a quite important protein in cell division and cell survival. If RAS is not longer negatively regulated, this leads to increase of proliferation and increase of survival of certain cells. And those cells divide in an uncontrolled manner and produce those tumors you can see on the skin of the patient on the right-hand side. The last example of the point mutation I would like to talk about is a disorder called epidermolysis bullosa. It is a quite rare inherited skin disorder which is causing permanent inflammation. This is quite evident on the patient on the right-hand side. Those kids are in constant pain and the skin is severely inflamed. So fractions are fairly common and the skin is always irritated. In contrast to the disorders I showed you before, this disorder can be caused by mutations in many different genes, mainly in genes that are in charge of producing proteins that are important extracellular matrix proteins. So we can have mutations in the keratin genes, but also collagen genes producing those symptoms. And the type of the mutation and also the number of mutations is indicative for the severity of the disease. For all of those genetic disorders I have showed you before, you need to appreciate the fact that unfortunately those disorders cannot be cured by a pharmaceutical intervention, and the patients affected by those disorders rely on the treatment of the symptoms for a lifetime, because most therapies are simply symptomatic. We are able to treat the symptoms, but unfortunately we cannot treat the cause using conventional pharmaceutics. Fortunately, In the last 2 decades, gene therapy has made incredible progress, and nowadays certain therapies become available even for the carriers of those mutations causing gene disorders. Gene therapy is a technique for correcting defective genes that are responsible for development of those symptoms. In most cases, a wild type or intact gene is inserted into the genome to replace the mutated gene. In order to achieve this, usually carrier molecules called vectors are used, and those vectors can be fairly different. In most cases, in gene therapy, viruses are used, but again, we will learn the details of gene therapies in a dedicated lecture later on. What I would like to show you here is actually something quite important and interesting, reported quite recently by the BBC.
A promising development in the battle against HIV. Scientists in this Philadelphia laboratory have altered the immune system of patients to make them resistant to the virus. The improvements were temporary, so Jay Johnson still needs to take daily medication to keep the virus in check, but he's delighted to have been part of the trial.
It makes me want to shout out to the world that, you know, there could be an end to this.
HIV attacks the immune system by locking onto proteins that poke up from the surface of white blood cells. About 1 in 100 people lack that surface protein, so they're naturally resistant to HIV. The scientists modified the DNA of the patient's immune cells to mimic that rare mutation, so the virus couldn't latch on. The cells were multiplied and re-injected into 12 patients. 6 patients stopped taking their medication. In 4, the levels of HIV decreased, and in 1 patient it fell to undetectable levels.
What we're looking at is not only a potential new therapy for HIV that still needs some time for development, but this is the first time this type of gene editing has been used in humans. So it can be applicable to many other diseases, genetic diseases, and so forth.
Improved drug treatments for HIV have transformed the outlook for people like Gary Bruff, who nearly died from AIDS 17 years ago.
There's literally no need for AIDS at all today. If HIV is diagnosed early and treatment's available, then AIDS is a non-event.
This is a long way from being a usable treatment, and it would be hugely expensive. But this small trial in the New England Journal of Medicine may point the way to research which can boost the health of those living with HIV. Fergus Walsh, BBC News.
In addition to the approach I showed you in this video, where a gene has been specifically removed in a patient cells, we can actually use more sophisticated strategies to treat previously uncurable disorders. And here, in this article published last year, the researchers use gene therapy and gene editing techniques to treat epidermolysis bullosa, which is the disorder caused by mutations in genes coding for keratin and collagen. Here, the scientist isolated cells affected by the disorder from the patient, then expanded those cells in a dish and introduced a healthy copy of the gene affected by the disorder in a dish, followed by expansion of those now cells carrying the correct version of the gene and transplantation of those artificial skin sheets onto the skin of the patient. And indeed, this therapy was really successful and cured the patient. So I hope that in those 3 lectures I indeed have convinced you that even as a pharmacist you need to know a lot about genetic disorders and cutting-edge gene therapy approaches. And this is something we are going to be having a closer look at during our dedicated lecture on gene therapy in spring term. With this, I would like to close the session and thank you for your attention.